Recording. So the review session, they found a room. Unfortunately, this building is generally locked up on Sundays. So they said Jamrich is available. So it's room 1318 that we will have a review session Sunday starting at 6 in Jamrich 1318. The outside doors should be unlocked, so you won't have any issues getting in. I'll send an announcement to the whole class about this because not everyone is here today. Shocker. Yeah. You think I don't know, but I know. I can tell. Okay. So, and I need to sit down and make a firm cutoff on the material for the third exam. It will certainly cover the slide set on light and the light reactions, all of that. It will cover the Kelvin cycle for sure, most likely the PCO cycle, and then I'm debating on where to put C4 and CAM. So I need to sit down and look at that. So I will be sending out announcements, hopefully by the end of today on that, as well as a reminder of where to go for the review session. So the review session will be questions and answers. I won't lecture at you, it's just come with questions, okay? All right, so we are continuing on with applied uses of hormones and how they can be used in commercial crop production or in your own home potentially. So ethylene has the action of promoting fruit ripening, but not with every fruit. And so if you're growing fruit to sell as a commodity, you do want to be able to have them hopefully ripen in large numbers all at once. And from the plant's perspective, of course, that fruit ripening, there's a whole lot going on in order to shift that fruit to be mature. And with the biggest issue is that the seeds have to fully mature and be ready to go. So very often the fruit won't turn ripe, so to speak, until the seeds are good to go. All right, so you have to, of course, not kind of force the condition too early. You do want the seeds in there if you're from the plant perspective, right? And so that's why fruits don't usually show any vivid colors until quite late. You know, you see the apple, but it might not turn red, or you see the banana as green, won't go yellow. And so thinking about those ideas from the plant's perspective. And then from our perspective, once it ripens, it's ready to eat. We generally don't like the taste of unripe fruit. The plant deliberately puts secondary metabolites in the fruit to make it taste bitter. And it's because they're not done yet. They want the seeds good to go so that who's ever eating that fruit will hopefully disperse those seeds into new habitat. Of course, this is big business in the food and agricultural realm. And so here you're seeing the difference of, so climacteric means that they are sensitive to ethylene. Non-climacteric means they won't really, nothing much is going to happen if you expose them to exogenous ethylene. So you can see that there's quite a few that are relevant in our food nutrition that are climacteric. And so they've known about this for a while. And for example, with apple orchards, sometimes they'll spray their apples as they look like they're getting close to maturity, and they want to just hire once to get people in there to harvest. They will sometimes expose the plants to ethylene, often through that ethafon chemical, in order to kind of snap them into the final maturation phase. So you can do that at home. I have to do that a lot. I'm sure some of you have noticed around here, you can get green bananas. You walk into the store and that's your only option, green. It's like, okay, if you do this trick, you can get them to turn yellow much faster and ripen up. Does anyone like really ripe bananas, like when they're freckled? Yeah. So some people really like that and a lot of people don't. It's kind of a personal thing. And one thing to be aware of, I try to eat when I choose a banana, I try to get it so it's yellow, but just having turned yellow. And with the idea that if it's kind of partially unripe, so to speak, it'll be more in the complex carbohydrate form. It won't be so much simple sugar. If you've noticed the difference in taste between a relatively, somewhat unripe and then one that's freckled, the sweetness value, it's very different in sweetness. And I would prefer to get carbohydrates in me rather than simple sugars. And so I tend to go for bananas that are somewhat underripe, I guess, in that regard. So just something to think about. But yeah, that's a good trick. I do that a lot. And then just remember that if you're getting into the plant's life stage where they should be producing flowers, you can actually manipulate that as well, where you might want to delay it for whatever reason. And that can happen, except with pineapples, a famous example where it actually stimulates the pineapple flower. But many, many other crop species, it would delay the flowering. So just be aware of that. It just depends on the species of plant. Pineapple, mango. By the way, although I've never grown one like that, those look amazing. This one I grew, the middle, the pineapple there, I grew in the greenhouse here. So this is a store-bought. You can see it's not quite store-bought level, but it was still really sweet and tasty. So you could do that yourself. I had to put Mr. Potato Head in there because I research with potatoes. So this is my windowsill in my office. So if you buy a store-bought pineapple, what you do is you can take off the top portion, which you always do anyways, right? And you can plant that. You can clean off most of the fruit on the bottom and just gently place it into a pot of soil and water it and let it go, and it'll start to grow. And that's what I did. And then you have to be patient though, because for the pineapple, they have to go through puberty, so to speak, before they will decide that flower production is good to go. And so I had that pineapple plant, I think, in the greenhouse for two years. And then after that, I put a large clear plastic bag over the pot and cut up some apples and threw them in there. And that did help a little bit with fruit production. So after the leaf or the flower was there, I popped it, and it grew tiny but tasty. So it's a thing that you have to do with some patience. So, but they're so cool. It's such a cool flower. It's a very beautiful flower. And then when the fruit starts to form, you can watch it, progress. It's just really fun to watch because it's such a striking fruit, very different than, what we have around here in terms of crop production. We don't do pineapple around here. So the plant can get quite big. So you need space in your apartment if you're going to do it, because they can get quite wide. So just be careful. I've had several people in the community call me up, hey, my pineapple plant's gotten really big, but I don't want to throw it away. So we usually say, yeah, bring it in. So keep that in mind. If you are moving at the end of December and you have a bunch of plants that aren't going to make it with you to your next destination, the greenhouse will take good care of them. We will keep it going. So yeah, be patient. I've had there, I don't know if you've noticed, I think it's on the third floor. We have a yucca plant in a big, huge pot on the third floor. And we got that from a family here in town. That yucca plant is over 30 years old now. So they had that when their children were little. All 4 kids grew up with that plant. They named it Igor. And so they were like, we can't, it's got so massive for their house. So I said that we would take it and take good care of it, keep Igor alive. And we've actually had Igor propagated. So he's had his own version of offspring cloning. And so just be aware that the greenhouse is a good home if the plants get too big or you have to leave them. Okay. So yeah, it's a fun thing to watch progress over first the flowering and then to get the fruit production. So ethylene is also famous for its ability to snap seeds out of their dormant. Now it's not that simple. Many things have to happen for seeds to decide to germinate, but ethylene can certainly be part of that process and help get that to happen. bud dormancy as well, sprouting with potatoes you see on the right. And so, you know, very often what you do when you get seeds is you'll wet them, and just that action can actually trigger a lot of hormone shifts in the seed, just getting it wet. And so it'll happen naturally. It's not like you have to expose them to, you know, like a cut apple to release ethylene, but sometimes they will do that deliberately in order to get all the seeds, you know, let's make sure they all germinate all at the same time. So they can use that in the commercial crop production. So if you want to apply it, it's often done through liquid production or liquid application, where you will use Ethifon, and that's a liquid, and then you put that into the irrigation and water the plants with that. And then the plant will take that up and run a simple pathway to make ethylene out of that. And so we can get that happening for if you want to speed up the fruit ripening. Maybe there's a frost coming along and you got to get your fruit harvested and in storage. You can synchronize fruit set and get it so that all your plants are forming fruit at the same time. Pruning, sometimes some people will do that with cotton, cherry, or walnut production. They'll use it to get it to, so cotton, by the way, is the fruit. We get cotton from the cotton fruit, which is weird. It's trichomes essentially on the seed surface, and then they weave that into cotton. It's kind of bizarre. Who thought of that? You can use it to keep your plants from not bolting, not getting too leggy. I've seen that done in certain cases as well. And then if you harvest your crop and you have to put it into storage before it gets to the supermarket or the farmer's market, you don't want ethylene in that stored room or container, because it's going to hasten ripening and over ripening before people buy it. So you want to keep ethylene low, which usually means you need to keep oxygen low, because remember, to get ethylene production, the plant needs a good dose of oxygen. So if you can lower that around your apples, for example, and then they often will pump CO2 into the container that has the harvested fruit. Cut flower industry, we mentioned already, silver is often added into the water that they're sitting in. But, you know, even with all the different things they can do to try to either keep ethylene synthesis low or just sop it up, kind of soak it up and get it complex so it won't react with the flowers, they still have quite a battle. There's a lot of cut flowers that get thrown away, so there's a lot of loss. So it's a big risk. They have to really know what they're doing. So you can certainly take the action of, well, if they make the ethylene, let's throw in the chemicals that will kind of sop it up and keep it from acting on the plant. So potassium permanganate is commonly used for that. And that's pretty heavily used when you have apples being stored. There's a new variety of apple that came out. Minnesota, University of Minnesota got that one going. Has anyone heard about it? I'm trying to think of the name. I think they named it either First Kiss or Sweet Kiss, something like that. First Kiss? Yeah. So I have not seen it in stores around here. That's one thing about apples around here. You just get the Main Street apples. There's all kinds of other apples out there. It's like, come on, where's the variety? Because the First Kiss sounded really good. And then I was at some potluck party, somebody around in the area, he has his own apple orchard, and he had some really rare species, hybrids. And one was based on Honeycrisp, which is probably one of the best apples I've ever tasted. So, and it was a Honeycrisp hybrid of some sort. It was really good. So I wish we could get more of that in local stores. Okay, so this idea has actually been done. They can do this, where here's the case of apple or tomatoes, where of course shipping ripe tomatoes is not really a good thing to do because they get bruised and damaged so quickly when they're already ripe. So then they get trucked thousands of miles. Doesn't work. So usually what they do is they pick them green and ship them green, and then they want to keep them green until you get to the store, and then or close to the store, and then you can do part two, so to speak. And so they've actually done genetic engineering of some of these tomatoes in order to essentially block ethylene synthesis. And so that's what this schematic is trying to show, where they use this bacteria called agrobacterium. And in my other class, I just came from my other class where a student in that class is trying to isolate agrobacterium from different galls on plants in the area. So we'll see if we can do it. But agrobacterium is a workhorse bacteria. It's heavily used in order to manipulate the genetics of plants because it's so good. It's a plant pathogen. It's so good at getting into plant tissue. And the amazing thing that it does, it has a plasmid. They call it the TI plasmid, tumor inducing. It actually inserts its plasmid into the plant DNA, and then the plant will express those genes. And so they know that the pathogen is doing that naturally. So what they do in the lab is they take agrobacterium and they take out the tumor inducing genes, get rid of that, and they put their gene of interest into it, and then allow that to infect plant cells, which is all done in tissue culture techniques. You're not working with a big plant. You have plant cells that you put into a broth and you get it shaking, and then you add in the bacteria and you get those cells to interact with each other. And agrobacterium will infect and insert its DNA into the plant DNA. So that's what they're trying to show here. And in this case, for this particular idea being shown here, it's a way to do antisense technology, they call it, where it essentially creates a complementary copy of the sense messenger RNA that would code for these enzymes to make ethylene. So if you make the antisense complementary strand, it will bind with any of the transcripts that are supposed to be transcribed for these enzymes. Your antisense will bind to that, and then it won't get translated into protein. And so you've effectively blocked the last step, that translation step. So the enzymes don't get made, but the plant will still respond to ethylene if you later say, okay, now I want the tomato to ripen up, and then you can spray it with ethylene. They have the receptors, everything's good, and they can ripen up. So this is something that has been done. I don't know if it's still mainstream. This did create quite a bit of controversy. People were worried about eating tomatoes that had been modified in such a way. So I'm not sure how much that this is actively done anymore. I don't know. Does anyone know? Yeah, I'm not sure. I'm not kept up with the laws can change so quickly, and I don't keep up with that part. So, okay, so there's some interesting applications of ethylene. Gibberellins, so that one can have some useful impacts on plants that you are growing for your business. So for example, it actually is pretty heavily involved in the transition of that plant from essentially puberty into adult. And so we tend not to say puberty in the plant world. We tend to say juvenile. They're in the juvenile phase. So if we say, oh, that plant's in the juvenile phase, it means that it isn't capable of forming any eggs or sperm yet. And then once you get it through that phase into the adult phase, it's capable. And so they know that gibberellins are definitely involved in that. So one thing they've done, and they got it to work with conifers. Conifers have a slow life cycle. It takes them a while to come out of their juvenile phase. It can take anywhere from 10 to 20 years. If you're trying to make profit, like let's say you want to grow Christmas trees, and you found a perfect Christmas tree and you want that Christmas tree to start producing seeds so that you can grow those seeds and get more of those perfect Christmas trees. Well, you would want that Christmas tree to give you seeds sooner rather than later. And so what they have done is they can actually spray the conifers with the gibberellin solutions, and it will actually speed it up and everything works out. It's like it does get seeds that are fully formed in the cones. It works if you do it correctly. And so they've been able to, so when you say the word precocious in plant biology, that just means earlier, much earlier typically than what is the norm. And so here's a nice picture where you have a cute little tree. I mean, that's a very young tree. It should not be producing cones at this stage of growth, and yet it is. So this one was giant sequoias. If you've ever been lucky enough to see the giant sequoias, takes them a while to grow. They're pretty massive. I was lucky enough to go see them at Yosemite National Park right before COVID. It was August before COVID struck. And has anyone been there, Yosemite, seen the giant sequoias? Highly, highly recommended. It is truly awe-inspiring to walk amongst the giant sequoia groves. It was just, I'm getting goosebumps just talking about it. It was really wonderful. So if you ever get that experience, highly recommend it. Here's white spruce on the left, showing the cone formation. So that's something that can get done in the real world. It's odd, but sometimes you can manipulate the flower where you can force it to produce male flowers with gibberellic acid, or you can, with corn production, you want more females than males because that's how you're going to get your corn on the cob is through female flowers. And so they will often spray to make sure that this female flower is developing. Gibberellins can also deal with apple fruit set. They also use gibberellin. I don't know if you've noticed with apples. Have you noticed some apples are very roundish and others kind of are elongated, kind of tall and a little skinnier, and not so just perfectly round. Apparently, at some point they did what's that called when you focus groups where you bring a bunch of people together and ask them their opinions. It's usually food related, like eat this, what do you think? And there's a scientist there taking all kinds of notes. They've done those focus groups, and apparently, at least for a while, at least a decade or so ago, people actually liked the fruits that were a little bit taller and not round. I don't know why. I never did see, they didn't describe why. But that's why they started spraying some apples so that they kind of grow tall instead of staying more squat and round. So gibberellic acid was used for that. I don't know if that's still being done or not, but and it does help stimulate seed germination. So I'll talk a little bit more about that. It can be used for getting all your seeds effectively germinated. Here we're seeing that idea again on stem elongation in the grape vineyards. So it's not really affecting the fruit directly. What it's doing is it's stimulating the vine and the stalks in the cluster of grapes to elongate, to grow the stem growth. And then that just spaces out the fruit better so that they can get bigger, and usually they get a lot sweeter. So that's the big payoff, is you end up with much sweeter fruit and can sell it at a higher price. That's always something to think about when you're in the grape production. There's the fruit elongation with apples. Citrus fruits, lemons and limes, they can actually show the correct amount of gibberellic acid. Exposing those trees at that time with the correct amount can actually slow down maturation of the plant and its fruit, and they sometimes will choose to do that if they're trying to keep their orchard going in production as long as possible, and to extend the market period, so to speak, so that they can keep supplying the lemons and limes throughout much longer growing season. Sugarcane is kind of an obvious choice if you think about it for a second. If anyone's driven next to a sugarcane field, it's impressive. I got lost in Florida one time driving on the back roads, and I'm like, I don't think we're in the right spot when we're on a 2-track dirt road and we're driving through sugarcane fields. You look to the left and the right, and it was like you were going into a tunnel of sugarcane. And we were just in a little Subaru, and the sugarcanes were way above us, and then some pit bull dogs are running after us, and like, I don't think we should be here. So, but the sugarcane field, I still remember that. It was impressive. And you can imagine if it's the actual cane, the stem that they're harvesting for sugar, you want good stem production. And we know from lab that spraying those dwarf pea plants led to really good stem production. So this is kind of a no-brainer to go ahead and apply gibberellic acid to your sugarcane. They've done some nice studies where you can get 2 tons more per acre of sugar. That's a lot. So that's certainly going to impact your bottom line on how much you can get out of that field. So another very lucrative way to apply gibberellic acid is for malt production in the beer brewing industry. And so what's going on here is the seeds, if you expose them to gibberellic acid, what it does is in the seed, it starts stimulating the alpha amylase enzyme. And what that is doing, do you remember what amylase attacks? What carbohydrate? You have it in your saliva when you eat carbohydrates. Do you remember what kind of carbohydrate? Starch. So have you ever done that trick where you put like a saltine cracker on your tongue and you just let it sit there? And it's, if you do that, the alpha amylase in your mouth will start breaking it down, and first it'll taste really bland, and then pretty quickly the sweetness kicks in, and that's the alpha amylase. And so if you're in the beer brewing industry, what you want to do is you want the starch in the seeds to be broken down so the yeast can act on it easier and quicker. Yeast have a really hard time attacking starch because it's a complex carb. So if you can break that down into simple sugars, then the yeast can feast very quickly. And of course, your beer brewing speeds up. And so the barley seeds can get an exposure to gibberellic acid in order for the malting step and all of that to go through faster. And then the yeast can attack the broken down products of the alpha amylase is acting on all the starch there. So we have the yeast involved in that, and then of course hops are often added. That's another plant to think about in the beer industry. And then of course the barley too. The barley as well. And so does White's Party Store still sell like beer making things? They have kits. They used to sell kits. Has anyone tried beer brewing? It's fairly straightforward. It's easier than wine, in my opinion, but fun to do. When I was a grad student, they knew I was in the microbiology realm, and so hey, do you think we could use your autoclaves to sterilize? Like, I suppose I could sneak it in with some of my other stuff that I need to autoclave. Because of course sterility is a big deal. You have to have everything really clean and sterile. So I got roped into that end of it. Probably the best beer we brewed was, it's going to sound weird, chocolate cherry coffee beer. I know it sounds weird, but it was so good. And guess what? The head brewmaster was a little tipsy when he made it. He didn't write it down. He was too drunk to write down what he put into it. We could have killed him. It's like, this is really good. And how'd you make it? Well, I don't know. So we never did get a recipe. Tried to recreate it. We never got back to that particular batch. So it was a one and done. So what they do is they take barley seeds and they grind them up a little bit. That's the malt. When they say malt, that's what they're really talking about. And that's full of starch. And so you want it to get broken down to glucose so the yeast can take that. And so adding in the gibberellic acid can get this step to really progress. So if you go on a beer brewing tour where you can go behind the scenes, you should ask them, do you add any gibberellic acid to your barley mixture? It'd be interesting to see if they know that. They might be shocked that you'll ask something like that. So big business, obviously, billion-dollar industry. And so they always say that Benjamin Franklin came up with that quote, beer is living proof that God loves us and wants us to be happy. I actually read that that's somebody else kind of did that, and this is what this is definitely a Ben Franklin quote, but this one there's debate that he might not have said it quite like that. That's kind of a different take on it. But here's your barley, and what they're trying to show here is that this is the baby seedling inside, and you have that endosperm in the seed, and so they're just trying to show that you can certainly have gibberellic acid coming from the tissue of the baby seed inside, but then what it's doing, and then of course the beer industry feeds gibberellic acid, doing breaking down the starch in the seed, and then once it gets simpler sugars, then the yeast start feeding on it. So that is this is just showing in words what we just looked at. And so in the natural world, the baby seedling inside the seed does have its own gibberellic acid, and it will start synthesizing it, releasing it to get the endosperm to start breaking down. Lots and lots of research going into seeds, how they get set and matured to go dormant, and then how to activate them and get them germinating. It's a big area of plant physiology. Here they're just showing the alpha amylase. If you treat versus you don't treat with the seeds exposure, so you can see that the enzyme, alpha amylase enzyme, really spikes quickly when you expose those seeds to gibberellic acid. So just a visual to get you thinking about that idea. So now on the other hand, sometimes you don't want gibberellic acid to really be cooking along in your plant because it'll produce an effect you don't want. So sometimes bigger is not always better. Sometimes you want short, squat plants. In the landscape industry or houseplant industry, you don't often want giant, tall plants because not a lot of people have room for that in their homes. And so, coming up, it's November now, you'll start to see poinsettias in the store fairly soon, probably. And that's a trick that they do. They don't, if you've ever tried to grow poinsettias, it's kind of a shame. So many people throw their poinsettias out after, they'll just like put them in the garbage. So I usually, if I have poinsettias, I'll put them in the greenhouse and just watch what they do. And the inhibitors will wear off over time. And so then they get really leggy. And so if you've ever wondered, like, what would it do, if you just leave it alone, they do get very tall. And so in general, we like the squat, compact poinsettias. They leaf formation and orientation just looks better to us, so we want that. So they actually can manipulate and try to block the synthesis of gibberellic acid. And so they do that for sure with chrysanthemums, which are certainly around right now being sold, and then poinsettias will be here pretty quickly, if not already. And so often it depends on the company and how they do it. It could be through genetic engineering, where they deliberately create a dwarf plant right from the genetics of it. So they can do that for production of certain grains, so certain cereals like wheat and barley. Sometimes they'll do that, and the main goal for that is you don't want the cereal grain plant to get too tall, because the harvesting machines aren't designed for that. And so if you want to easily harvest those seeds or the grains off of the plant, you want the plant to stay upright. You don't want them to get too tall and fall over, like the foolish seedling disease we talked about. And you don't want all that energy that the plant is doing through photosynthesis. You don't really want it to go into stem tissue. That's not what you're trying to profit off. You're trying to profit off of selling the grains. And so that's another reason it gives you less stem tissue, the less photosynthesis going into that. And it also makes it so that your plants are stocky and pretty good stock. They're not weak, and then you can easily harvest with your machines. So here, just looking at this idea of gibberellic acid, and that they actually have gibberellic acid inhibitors that you can use if you don't want the plant to get too tall. And so that is, you can do that through an external spray, like what's being shown here. So it's not through a genetic manipulation, or in this case, for example, these are wheat plants that were genetically engineered. And you can see that the far left one is your wild type, and then the other three are various types of genetic engineering that occurred, and looking to see how they would grow to maturity. So that last one is definitely a little too small. I don't think the harvest machines would work with that well at all. But probably the second to last one is good. So they're definitely looking at this idea, limit stem tissue amount. Hopefully then more of the sugar production will go into more of the so-called cereal grains, which are actually fruit. It's dry fruit. So genetic engineering is getting to be more precise and easier. And so one of the strategies for that is to essentially get extra copies of the gene that codes for the enzyme that breaks down gibberellic acid. So essentially the plant still makes gibberellic acid, you just put in extra copies to get the enzyme needed to break it down or metabolize it. And so obviously if you keep chopping up gibberellic acid and breaking it down, it's not going to be effective in the plant. So they're actually looking at doing this for turf grass on golf courses. Can you imagine if you're the one who gets this and patent it? You're going to be rich, rich, rich. So I don't know if they've done it yet. I know they're working on it, but you think about all the turf grasses worldwide, and you could go to those the golf courses and say, I can guarantee you'll never have to mow that. You're going to be rich. I think that's an awesome idea, and not having to mow, all the pollution from mowing. So if anyone knows about that or finds out about that, let me know. I think that's an awesome idea. Drought tolerance is a big deal. Crop production, that is probably one of the biggest abiotic impacts on your bottom line, whether or not you're going to have a rainy season or dry season. So having plants be capable of dealing with drought, we know that abscisic acid is one of the main hormones the plant uses to signal up into its leaves, hey, we're under drought conditions, we need to close down our stomata. And so what they've actually done in the labs, playing around with this a little bit, if you spray plants with abscisic acid, it actually kind of primes them to be drought tolerant compared to plants that did not get that spray. And so it's almost like you're getting them to exercise that trait and strengthening that trait. So they have been looking at that. The problem is that it's expensive. When they do the calculations of what would this cost on a more commercial scale, it's not really feasible. And so what they've been playing around with a little bit, there's this synthetic chemical that's close in structure to abscisic acid and seems to work pretty well. It seems to bind pretty well to the abscisic acid receptor protein. So it's like a mimic compound that the receptor is okay to interact with. And they're actually finding, at least in small scale studies, that it is actually better. It seems to kick the plant into the drought response very well. And it can be a very quick response as well. So you know, that's always a good idea when you know that, you know, isolating and selling abscisic acid is a pricey proposition. Then go ahead and try to find a mimic. So lots of effort going on with that. And with just getting the plants primed so that they're more responsive, more sensitive to drought conditions and can tolerate it better. So here I put in just the scanned the front page with that opabactin chemical that they've discovered. So that was just a couple of years ago from a group in Spain. Tenfold greater activity. So this is a big area of research looking at how to better, I guess, how to help the plant become more tolerant and more capable of surviving through the different seasons of drought that are going on. Okay, let me see. There's one other thing I wanted to point out if I can find it. Stop share, hang on. Whoops. Where did it go? Oh. So, just wanted to show this. Oh, I gotta exit out of it to share. Hang on. We were talking about pumpkins. So going back to that idea. So the pumpkin, I am always amazed when, if you've ever grown pumpkins in your garden, they have these beautiful flowers, very rather delicate flowers, and yet that can morph into that very tough pumpkin. So here you have the female flower and the idea of the egg production in the base. And so what you're thinking of is you got to get the pollen onto the female flowers because the pollen has the sperm, of course, and then you get the fertilization. And then once that happens, that is the stimulation to kick into fruit production. And so I know that people carve pumpkins, and I've been doing that for Halloween, but just remember, pumpkin is super healthy. It's not just for making scary caricatures. Is that right? Caricatures? Wow, that's a hard one. And so, you know, keep that in mind. It's got a ton of very, you know, the carotenoid pigments are very healthy for you. There's other secondary metabolites. So, and then the giant pumpkin idea we talked about, how, I mean, it's just kind of mind-boggling how massive they can get. So yeah, it's a thing. It's worldwide. That's the pumpkin regatta I was talking about. This is on the East Coast. Ooh, someone even put corn up there in their, maybe that's their backrest. They rest against the corn. But we should do that right in the Lower Harbor, right? You'd have to probably put a wetsuit on, yeah, but I think that'd be awesome. I don't know if this will. Oh, I can, if you want to watch this, I don't know if it'll do it. It's not letting me. Oh, well, you can watch it. I'll put this in the slide sets and you can, it shows like sped up pumpkin growth. So it's kind of fun to watch that. And then this one is a little podcast, like 5 minutes of the physics of giant pumpkins, if you're curious. So I'll post this. I just wanted to, my favorite one is the regatta. I think we should do that here. That'd be awesome. All right, so we'll stop there. I'm going to get into another topic. And I think we're into secondary metabolites and how plants defend themselves. So we'll start that on Monday. So hopefully I'll see a good portion of you at the review session on Sunday.